All right, so at this point, ladies and gentlemen, what we have done is we have looked at um, up to the base pairing. So we looked at base pairing, Chargaff's rule, which didn't tell us exactly what the structure of DNA was, but what it did do was that it did give us some hints as to what is going on internally with DNA. That being said, as always, the next scientist, I guess, if you will, to come along was an individual by the name of Rosalind Franklin. And why is there we go. So 1950s. This is in England. There is a British scientist by the name of Rosalind Franklin, and she starts studying DNA because, like all the biologists at the time, were studying DNA. She is using a technique called X-ray diffraction. So what she basically does is she stretches out DNA fiber into a thin glass tube so that most of the strands were parallel. Side note, I would like to say that she did do this during the metaphase of meiosis, because I'm sorry, of mitosis, my bad. Um, because at that time it is the DNA is already sort of lined up, and then she just had to sort of like add an agent into it to sort of unravel them and stretch it out. So it was actually a very awesome technique, in my opinion. She then basically took an X-ray picture of it. So much like if you take an X-ray of like your arm, you can see the bones inside of it. What an X-ray does is when it takes a picture, it will not be able to go through the densest part of material, which is why it's great for taking pictures of bones, because it can, it doesn't go through that dense material. So when she did this for DNA, the picture you see on the bottom of the screen there, that is the famous photo 51. And what we see there is it almost looks like a mirroring effect. Okay. What this tells us is that DNA is a coiled structure. That super important. And again, without this photo 51, I don't know if necessarily the structure of DNA would have been solved so quickly. So this X pattern basically shows that the DNA is basically twisted around each other. This is mathematically called a helix. Like there's like, oh, there's pentagons and octagons, and there's all these names and geometry for stuff. So if you have a coil that twists around itself, like mathematically speaking, we call it a helix. If you look at the dark spots on the top and the bottom, those are your nitrogen bases, and we can see the pairing. So remember, Chargaff did his rule about base pairing, and he said, okay, well, adenine and thymine always match, cytosine and guanine always match up, and we can literally see that in this structure now. We can see that each side has a mirror effect. Each side has a pair, like your left shoe, and then you have the pair, the right shoe. We can see that in this structure. I do want to say this just because, as a woman in science, this is one of those things that has kind of always irritated me. Rosalind Franklin clearly was a woman, and obviously this is the 1950s. And we all know that minority populations back in the 50s were really, not really acknowledged for the contributions that they made. That being said, this picture, photo 51, which is the key piece of evidence that eventually is used to determine the structure of DNA, was literally stolen out of Rosalind's lab. And originally, she never got credit for this. So, anyways, little moment of history, the history of women in sciences, of being overlooked by dudes, but that's okay. Hashtag bitter. All right, moving on. So Watson and Crick, dudes, um, were at the same time, we're also trying to understand the structure of DNA. They ended up building 3D models because remember, everybody is trying to figure out how it is that DNA does its three functions. The three functions of DNA, of course, being storing the genetic material, copying the genetic material, and then putting that genetic material to work. Either in the organism or the next generation, and everybody believed that the key to understanding the three functions of DNA was to understand the structure of DNA. Because if you know how something works, then if you know how something works, then you know why it works. Um, the thing is, they built model after model after model, and none of them were actually able to explain the DNA's properties. Now, it says Watson saw photo 51. That's not exactly true. Franklin's lab partner, who hated her, stole her picture and gave it to them. So there you go. There's the real history of it. Um, and when Watson saw Franklin's photo 51, he was able to notice those clues that I pointed out earlier, that parallelism and how that works. So he was able to figure out some specific properties of that DNA. Using the data in Franklin's X-ray pattern was what was able or enabled Watson and Crick to build the model that explained the properties of DNA. By the way, the Watson-Crick model is the model still used today. 50, 60, oh my God, I seriously can't do math right now. 70 years later, we're still using the same model because it works. Um, that same year, 1953, Watson and Crick publish a paper where they give Rosalind zero credit for any of it. Like I said, hashtag bitter. Um, and that paper that they published, which was a double helix, which was explained DNA as being a double helix. If you make DNA into a double helix, it literally explains every single property of DNA, which is why it was a very powerful thing. All right, so here is the double helix thing. What a double helix looks like, think of a ladder that gets twisted up on itself, okay? So when we say double helix, it is because it is two-sided, like a ladder. You got these rungs in between, and then it sort of twists itself up into a twisty, almost like a spiral staircase, if you want to think of it that way. Anyways, so again, when we say double helix and we talk about anti-parallel strands, double helix basically is a twisted ladder. If we look at this like a twisted ladder, it will explain all of the data in X-ray in the Franklin's X-ray patterns. It also explains many of the properties of DNA. This double helix model also explains Chargaff's rule, and it explains how the two strands are held together. So, a lot of these things that we know about DNA can easily be explained if I create a double helix model of DNA. And again, our purpose here as scientists was to understand not just what it looked like, but how does the structure help DNA function in the sense that those two things are always linked in biology. Structure and function are one of the guiding principles that we have always noticed in biology. All right. I'm sorry. I'm trying to bring up something else on my other screen. All right. So, if you're looking at the ladder, and again, anytime you have a ladder, you have sides of the ladder, and then you have rungs of the ladder. So, here is my very basic ladder. All right, so these sides of the ladder before it gets all twisted up, and you can see those in red now on my screen. Um, that is what we normally refer to as the backbone of the DNA. The backbone is made up of the sugar and the phosphates. You guys remember we talked about the nucleoid, the nucleotides that build up DNA, and how it was a sugar group, a phosphate group, and then that nitrogen base. Well, that sugar group and that phosphate group are what make up the backbone, the sort of the that part of the ladder. In the picture that you're looking at on your screen and possibly in your notes, that would be like sort of that blue ribbony looking thing. That would be the backbone. Okay, these rungs, which I have drawn in blue, these rungs are my nitrogen bases. So again, the sugar phosphate is the backbone, which I have drawn in red, and the nitrogen bases are I have drawn in blue. Now these nitrogen bases, they are paired. So like one side might be an A, and then of course the other side would have to be T. Maybe this side is C, which means the other side would have to be G. And then down the center, these guys are bonded together. Now remember, we really only know of three types of bond. Well, I mean, we know of a couple more different types of bonds, but the three main chemical bonds that we know of are hydrogen bonds, covalent bonds, and ionic bonds. So, looking at this, oops, oh, gotta go back to this mouse. There we go. Oh, I can delete this. My bad. All right. So when we say anti-parallel strands, what does that mean? That basically means that even though they are matched up and they are next to each other, think of one as sort of being upside down compared to the other. Okay. They run what we call anti-parallel, and that is actually really important because that means that the structure is going to stay stable throughout the entire molecule. And again, these are really, really, really long molecules. So stability is actually extremely important because if they aren't stable, if they're not matching up properly, then that is going to be problematic. All right, so they go in opposite directions. So you can see again here, you have one arrow pointing up, you have one arrow pointing down. They are anti-parallel. The DNA strands run in opposite directions. That was what anti-parallel means. What this means, though, is that the bases can come into contact with each other near the middle of this 3D molecule. And again, they carry their sequence of nucleotides arranged like the four-letter alphabet. So you can see the C and the G together, A and T, C and G, C and G. But over here, the C's on the other side. Again, always paired up according to Chargaff's rule. And that is how they come into contact with each other. All right. So double helix hydrogen bonding. So between the base pairs, we have hydrogen bonds. Okay, hydrogen bonds. Hydrogen bonds are always forming between the base pairs. Now you might be like, well, wait a minute, isn't hydrogen bond like the weakest of all of the chemical bonds? Well, yes, as a matter of fact, it is. And then people are like, well, then why do you have the super weak bond holding together the most important molecule for all life? Remember, one of the functions of DNA, not only does it have to carry the genetic information, but it has to be able to copy that genetic information. This is just enough force to hold the strands together, but it's weak enough where they can unzip and then get copied. So it's absolutely freaking brilliant, okay? In the sense that this bond holds it together, but it's weak enough so that they can come apart for copying purposes. I do want to point out that the backbone is just like your backbone is much stronger, and the backbones are held together by covalent bonds. But the hydrogen bonds go down the center and they hold together the nitrogen bases. The hydrogen base, the nitrogen bases, excuse me, down the center. All right, so adenine and thymine, you can see there are these two little bonds between them always. Guanine and cytosine always have three little hydrogen bonds. One other thing that I want to point out, and I don't know if it's necessarily in your notes, but you may want to add it. You also will notice that A's and G's have these two ring structures. These are the pyrines, and these single ring structures are called pyridamines. Now, ladies and gentlemen, I want you to notice that it's always a single structure, a pyridamine, matching up with a double ring structure, pyrene. Why? Because that way the center is always 1, 2, 3 circles wide, regardless if it's a C and G or an A and T. And again, that three uh, circle structure down the center gives it stability. Okay, this creates a perfect fit. This means that this molecule isn't going to be wider in some areas, skinnier in others, because anytime you have differentiation, it has a weaker structure molecularly. All right, so that again, the double helix, hydrogen bonds hold it together down the center. Covalent bonds hold together the backbone. A and T always have two bonds between. C and G always have three. And if you look at the actual bonding, we have a two ring structure, always connected to a one ring structure, regardless of what that might look like. Whether it's C and G or A and T, I always have three rings across. All right. So I think this is the last part of the notes. Hydrogen bonds with thymine, always adenine. Hydrogen bonds with cytosine, always guanine. This goes back to Chargaff's rule. Does this make sense that a molecule is important and DNA is held together by weak bonds? Absolutely. DNA needs to unzip to copy itself, right? So again, one of the functions of DNA is that it has to be able to copy itself. If the two strands were held together by really, really strong bonds, they might not be able to unzip, or they might only unzip partially, or maybe the zipper would get caught. All right. And again, we already know this because DNA has to be able to separate for copying purposes. I think that might be, yep, that is the end of the notes. Awesome. All right. Any issues or questions about the notes?